we're moving from G1 into S. This checkpoint confers an ability to cell, as do all other checkpoints. Right? So, an organism developed this mechanism, and then that mechanism turned out to help the organism. Specifically, this is going to make sure that replication only occurs once. And it only occurs when a cell is ready. So, we know that the S-cyclin CDK is going to drive this into synthesis. We also know that the P53, P21, check 1, check 2, that pathway can arrest S-cyclin. So we know that S-cyclin drives us forward. We know one of the mechanisms by which this is going to be shut down. So on DNA, there are multiple segments, and those segments are where we start copying, where the body starts copying the DNA. That segment, that linear sequence of DNA, T's, A's, G's, and C's, is called the origin. There are multiple origins along DNA. So replication is going to start many places on DNA at the same time. So for example, this might be an origin. This might be an origin.
What determines, is it just like the structure?
It is the sequence of DNA.
Is it on both strands?
That is a very good question. I don't know whether this protein binds to both strands. I assume it does. All right, the long story is that at the ORC, the origin of replication complex, a protein binds. That protein binds to these basically adapter molecules, CDC6 and CDT1, which go ahead and recruit MCM, code name for helicase.
Okay.
What does helicase do? What?
It like opens it up.
It opens what?
DNA.
Okay, very good. All right, so ORC binding to this origin ends up recruiting helicase. The important part of this story is that this structure is assembled here in G1. So in G1, we have the adapter, the origin, and then helicase. Have we started copying yet? No. The transition to S is quick. The SCDK is going to basically inhibit the two adapter proteins. Do you see them coming off here? You might not want to write this first. You might want to pay attention first and then write later. Plus you have this diagram, right? So don't forget that. Okay. Once these adapter proteins are inhibited, MCM recruits the rest of the replication machinery, and that's going to include the polymerases and the primases. Do you guys remember that whole complex deal?
Which one is the one that makes sure that the DNA is instantly boiled?
Topoisomerase?
And what is that name in prokaryotes?
Gyrase?
Yeah.
Those are really specific questions.
I know, I forgot that.
No. Where's my replication video? Ah, here it is. All right, so this complex. Is going to be, lost it. What's assembled here? Now, the SCDK is also going to phosphorylate the ORC. And what that is going to do is prevent rebinding of these inhibitor or these adapter proteins. Sure. So in the previous slide, we saw that these adapter proteins bound to the ORC. That recruited MCM. Then the S-cyclin CDK phosphorylates these and inhibits these adapter proteins. When this is phosphorylated, it cannot bind to the adapter proteins again, meaning that this complex cannot be assembled again. Meaning that once this is phosphorylated, this piece of DNA, or this part of DNA, is not going to be replicated again. Yes?
Why does the ORC split?
That's a very good question. I do not know the answer to that. I don't know. I haven't looked into that mechanism. What's important about this story is that the machinery is assembled in G1. It is triggered in S by the S-cyclin CDK complex. And once triggered, it is turned off. So, this cannot reassemble. DNA cannot replicate again here. Let's go over the important parts again. This whole machinery, this machine, is assembled during G1. It waits, it's dormant. It can't do anything until the S-cyclin CDK concentration is high enough that it inhibits the adapter proteins, triggering replication. In that process, the ORC is phosphorylated and replication cannot happen again. What's that? Because of this phosphorylation specifically.
It's like a change in structure, a change in
Same idea back to Bio 1,
Yeah.
like third day.
So it's phosphorylated after or before it replicates in DNA?
It's after the...
So like it replicates and then is phosphorylated?
more think of it as MCM is released as this is phosphorylated. So it's like, as it leaves, as it begins to replicate, it's phosphorylated. Or it's phosphorylated as it begins to replicate. All right, you can stop that one.